Welcome to the awards and plenary session. My name is Amina Pollard and I have the distinct pleasure of introducing the award presentations today. Please add any questions for the speakers to the chat. The recipient of the 2021 Raymond L. Lindeman Award is Dr. Ryan Lepak, who is currently a postdoctoral researcher funded by the U.S. National Science Foundation and Cornell University and stationed in the Great Lakes Toxicology and Ecology Division within the U.S. Environmental Protection Agency. This award honors a young scientist for an outstanding peer-reviewed paper in the aquatic sciences and is given in honor of Dr. Lindeman, whose seminal paper on trophic dynamics was published just after his passing at the age of 27. The 2021 Lindeman Award is given to Ryan Lepak in recognition for the paper Mercury source changes and food web shifts alter contamination signatures of predatory fish from Lake Michigan, which was published in the Proceedings of the National Academy of Sciences of the United States of America. The essence of Ryan's study was to understand how two major drivers of change, rapidly changing mercury emissions inventories and invasive species-based food web perturbations, interact to alter methylmercury sources and bioaccumulation pathways in aquatic food webs. To do so, the authors used a novel multi-tracer approach to sample two independent archives from Lake Michigan to examine synchronous responses over a 35-year period. This study challenges three prominent mercury paradigms. One, that global emissions are the dominant mercury source, comprising atmospheric deposition in the Great Lakes region. Two, that decreased loading would necessarily result in decreased fish burdens. And three, that mercury isotope markers of water column sunlight penetration versus measured increased water clarity would be paired. I defer to Ryan, of course, to share the specific findings from this study, but I will note that the results of this paper and the innovative methods to determine mercury sources developed therein will prove to be critical to better understanding the sources and dynamics of mercury in aquatic habitats and organisms. This study contributes to the legacy of Dr. Lindeman by considering an integrated food web compartment approach and by using multiple tracers that represent different facets of this food web. Please join me in congratulating and welcoming the 2021 Raymond L. Lindeman awardee, Dr. Ryan Lepak.
Right. Thank you, Amina, and thank you to the entire board, for the nominations board, for this selection. I have to admit I was very humbled to receive such an award. Looking at such an esteemed list of recipients in the past, it's truly remarkable. Not too different from raising a child. They say it takes a village to raise a child. To raise this academic scholar, it took many teams. And so to start, I think it'd be prudent to thank those individuals. We've got three major coalitions that I've worked with to work on this paper. We've got those at the EPA and not only my nominator, but current mentor here at the EPA, Joel Hoffman. And then separately, Beth Murphy over at Water Compliance. My PhD mentors, and I say two, Jim Hurley at Wisconsin and Sea Grant. And then now on his fresh retirement, Dave Krabenoff at the USGS. And of course, the stellar team at the USGS, because this is a very large team. Thanking those that I love, my family, and let's get into the science. So we had a remarkable problem that we were tasked to solve on the onset of this project. We've got, through time, we know that there's been declining emissions, declining concentrations of mercury, both in the atmosphere and through the wet deposition to the lakes, to the Great Lakes. Wet deposition is the prominent source of mercury to these lakes. Yet we're not seeing synchronous decline in concentrations across lakes that are considered highly remote and pristine, like Lake Superior. And we're not seeing them in more urbanized regions like Lake Michigan. Thankfully, the US EPA, through the Great Lakes Monitoring and Surveillance Program, as well as its complementary piece at Canada, had the foresight to start collecting specimens as early as 1975. So this gave us an opportunity to bring new tools to old fish. A lot of things have been happening in these Great Lakes through time. For instance, a lot of these lake trout I've measured are size standardized. But through time, you'll see that they're progressively aging in some lakes and not in others. Of course, we don't account for that in our study, but others have, and we found a really confusing story of sometimes declines, sometimes breakpoints and steady points, and in other instances, increases through time in this short time period. And so you could see a certain scenario or a motivation here where this set of information could be used as maybe ammunition to reduce mercury mitigation strategies domestically in going forward, particularly in the previous administration. So we were interested in demonstrating for another reason why these mitigation strategies are indeed working. If you're interested in the differences between Lake Michigan and Lake Superior, I got a selfish plug here to encourage you to enjoy and attend tomorrow's stabilized tip session near the beginning of tomorrow's final day. So what does mercury just look like in these Great Lakes? Well, it's amongst the lowest in filtered methyl mercury in the world. In fact, often lower than the open Pacific and often lower than that found in the Atlantic. So we're talking about, in terms of methyl mercury, very squeaky clean ecosystems. Yet we have fish that are often above consumption thresholds. To understand why that is, you kind of have to understand how methyl mercury enters the food web. And this is really where we pitch in the Lindeman concept. And it was surprising to me when I started reading about this more and more, just how much foresight this approach has, right? So when we think about mercury, we think about how mercury enters the food web through the basal components. We're talking about the algal cells. And of course, the anion or the ligand attached to this methyl mercury is really important to how efficient that process is. So all of this is interacting, much like how Lindeman described it. You know, he had a series of nutrients, and here we're going to include DOC in that. And that DOC is interacting both with the bacteria for sustenance and with the phytoplankton. Both create it and utilize it. Of course, the bacteria are forming this methyl mercury, so that's where that becomes highly important. And then the uptake of that occurs in the phytoplankton. So all of this is interacting kind of how Lindeman had described. And then to bring us full circle and why the fish are indeed so high, there's a process of biomagnification, and that renders the predator swimming in the water about 25 million times higher than the water they reside. Of course, this scenario I show you is really, on the right-hand side, is really a story for inland lakes and rivers. We've got these huge macro carbon molecules that are, allochthonous in nature, more complex and larger. And when we start to think about how that influences uptake, we can use something kind of like partitioning constants or something like bioaccumulation factors. And here we use bioaccumulation factors, and ecosystems have a predisposition to fall on this curve. And think of that as an efficiency of transfer to the food web. So in inland lakes, we're talking about maybe moderate efficiency. And when you move to the ocean, we lose a lot of these really large autochthonous carbon, and the efficiency increases. Of course, these algal cells are not necessarily needing that chlorine or bromine anion for sustenance, and it's not like dissolves like. And so it's not the perfect storm to create even higher efficiency of transfer. But the Great Lakes are that. And in fact, I'd pitch Great Lakes globally fall underneath this scenario. We've got, very low carbon content of autochthonous carbon that is comparatively simpler to bring into these cells, and therefore increases those contaminants of potential to bioaccumulate. And that really matters, because once you start to look across DOC windows, whether it's concentration as shown on the x-axis, or quality when you compare Minnesota streams, which is more allochthonous carbon, and the comparatively autochthonous carbon of the Great Lakes, you see that little differences make huge returns. And so, for instance, Lake Superior is about 5 times more likely to bring in methyl mercury to the food web than the lowest DOC streams in Minnesota. And that matters because that immediately sets the baseline for the food web. And here in the Great Lakes, we see that Lake Superior is the highest, and then Lake Michigan, Lake Huron are about similar, and then those are higher than Ontario and Lake Erie. Notice the color schemes. I'm going to try to keep those consistent when we talk about multiple lakes. And ultimately that matters because that impacts fish burden. So as that bioaccumulation factor increases, so does the net fish burden mercury. But what about Lake Michigan? Let's focus on Lake Michigan. Well, how has Lake Michigan changed? It is changing. It's constantly changing. And what's different from now and the past? In fact, that's really important to understanding the story. So in about 1995, there was an invasion of zebra mussels, which was considered highly detrimental to the ecosystem overall. And it wasn't until the quagga mussels invaded in about 2000 did we learn how much more impactful this scary story could be. So on the top panel, you see quagga mussel density through time every 5-year interval. And on the bottom, you see the zebra mussel density. And what you see is a complete reversal of the populations. And that matters because these quagga mussels are capable of filtering immense amounts of material from the lake and have taken a lake that was on the border of mesotrophic to something that's highly oligotrophic. In addition, we've lost one of the major food components of the food web, and that's the Diparya, which then feeds the primary bait fish of interest to lake trout, that's the alewife. So we've kind of shunted part of the food web. Then, at the same time, in comes another invasive species, which has become even more prominent in the diet. Although it's not the dominant component of the diet, it is more prominent. So when we think of all these things that are already happening in the ecosystem, before we even get into the tracers, we see that there's a lot of complexities arise. Naturally, that could impact mercury in the Great Lakes. Now, typically, the repository to understand mercury deposition in any given lake ecosystem is to take sediment cores. These are our typical archives. And what we learn here is indeed, as we've removed mercury from the ecosystem, the cores have responded and mercury is decreasing. But often this is kind of more the residue, and it's a big mix of sedimentary particles, both from the near shore and sedimentary particles that are formed in situ, and those deposited directly. So does that mirror or tell us really what's happening in the lake as it pertains to biota? Because ultimately it's fish is the sentinel that we care most about and why we study methylmercury dynamics. The study design is a little bit different than many of you may be used to. Any single given data point is comprised of 5 equally sized lake trout, and any box plot is comprised of 3 to 5 measurements of those composites. So sometimes a box plot will represent 25 fish. So starting early on, the color coding here represents sites where we have Saugatuck as a nearshore shallow, we have Sturgeon Bay as a deep, and a Charlevoix that's kind of intermediate. We see the concentrations are high, and as we started to clean up the ecosystem with the, as the Clean Water Act got a little teeth, we saw concentrations rapidly decline, which we're happy about. And then something happened. And of course, we've already kind of alluded to this being related to this quagga mussels and the zebra mussels, and you can see how contemporaneous that is with those insults. So let's look at just the bulk carbon in these lake trout and what's happening. Well, until about 2000, these values are fairly stable. And then slowly they started to increase. And we're talking about subtle increases, but increases nonetheless. What this suggests to us is an increased degree of benthic reliance. Additionally, what we learned from this is that for some of these time periods, populations collected in the north and collected in the south are actually distinct. And this has been also shown further with marker studies and tag studies. This is also supported by the decrease in nitrogen through time. So following about 2000, nitrogen values started to decrease, again pointing or hinting towards benthic reliance or presumably more nearshore reliance. Now this is really where we make the most hay for our research here. Following this axis, this 202, we're going to just kind of consider that the source of mercury to fish. And in about 1989, 1990, we started to see the values of these fish rapidly change. In mercury isotope values, these are large changes. And those are contemporaneous with the removal of medical waste and mercury in medical waste and batteries. Continued improvement has progressed as we implemented the mercury air and toxic standards rule. So here we can demonstrate very clearly that our mitigation strategies here locally are improving fish. Likewise, we learned that the mercury must be coming in through the atmosphere because we've previously pointed to populations of fish that are distinct. And yet we're seeing a unified shift, a synchronous unified shift in the fish values through time. So that's why we propose this is from an atmospheric transport. When you compare it to the sediment cores, because that's kind of where we started, we find that had you only looked at sediment cores, you would have found the same direction, of course, but the magnitude had been subdued. In fact, sediments only reflect 25 to 35% of the magnitude and shift for this study. And as a teaser, Lake Superior also does this, as does Lake Erie from preliminary results. Now, Amina spoke to us already about water clarity in 199 and how, or in this other axis for photochemistry and how those might interact. So when we measure this value in mercury in fish, and we compare to Secchi depth, they kind of linearize nicely. So clear water's higher values. These fish were all measured following 2005. if we measured fish before 2005, we would have expected Lake Michigan to fall about here. So we're talking about really large changes. And this created a really fun conversation during the preliminary exams, because of course we all expected in that room to see a major increase in 199 following quagga mussels. It was very intuitive. And we couldn't have been more dead wrong, which was really fun to learn from. So on the line plot you see in there, that's Secchi depth, and it's kind of crazy because as the line goes up, the water is becoming more turbid, and as it goes down, it's becoming progressively clearer. And interestingly enough, this water quality marker in mercury values actually follows that. And this is really the nail in the coffin for us to demonstrate. In fact, these fish are swimming deeper into getting their food, and that's why the response is opposite of what we expected. So biology here is trumping the photochemistry. Additionally, both the nearshore and benthos are now a zone of intense nutrient recycling as a result of both cladophora reproliferation and the live quagga beds that are now present everywhere. So now we've mirrored, not only bringing a game fish closer to these regions of methylmercury production, but also enhancing that production to exacerbate the issue. So what's next? Well, we'll talk about a little of that tomorrow, but we're looking now at understanding more complex compound-specific measurements, looking at both essential and non-essential amino acids, essential and non-essential fatty acids, to better understand how the basal food web impacts these fishes through time and space between lakes, and how we can better understand trophic dynamics therein. So I thank everyone for listening. I've got a series of funding up here that we should always acknowledge. The ASLO crew, again, thank you, Amina, and the awards committee, and those of you listening live or that are going to tune in later for the recording. And most especially, thank you to EPA Glenpo, who's given me unparalleled, awesome access to this wonderful repository of samples, and the Lake Guardian, which really taught me how to do research in these Great Lakes, inland seas.
Thank you so much, Ryan, for that excellent presentation. If anyone with questions could add them to the chat, that would be great. Quick question. What would have been the outcome without the reductions, presumably without the reductions in mercury emissions?
Excellent, thank you for that. That's a great question I skipped over. So concentrations today are not markedly lower than they were a decade and a half ago. And that's disappointing because we've almost halved the atmospheric concentrations. Had we not done that, it makes sense to assume we would be looking at concentrations in these fish that are double or triple what they are today. But because of invasive species, the rules governing the mercury that reaches fish and fish burden have changed. We're no longer playing by the same playbook. And so our expectations need to reflect that. And that's been a very contentious topic to discuss, but one that's really important.
Thank you. So does that mean that, oh, a question, what remediation steps or actions do you recommend going forward? What was I about to ask as well.
Yeah, thank you, Laura. So I think we've, in a really brief period here, we just quickly showed you a lot of slides and a lot of information. And I think what we've learned is that we're only as strong as the monitoring we're doing, and continued monitoring. Now, you can envision a scenario going forward where Minamata and the US continue to improve, Minamata Convention being the reduction of mercury globally, continue to improve emissions globally, where we continue to decrease concentrations. And truly, if we want to monitor that, at least in the sense of lakes, Great Lakes globally create an excellent sentinel because of this unique predisposition to be highly sensitive to mercury uptake. And so what would I recommend doing is continuing to monitor those ecosystems that are both highly important to the planet in terms of freshwater and sustenance, but also highly important and sensitive sentinels to mercury changes therein. Great. And do you know anything about non-microbial mercury methylators? I'm going to have to punt on that one. We have a really wonderful colleague, Sarah Jansen at USGS, and I would recommend asking her those questions. She's absolutely stellar at those things.
Fantastic. Thank you so much, Ryan, and congratulations on the Lindeman Award.
Thank you for your time.